Hi, my name is Miles and I'm here to talk to you about polymerase chain reactions, or PCR for short. PCR is in the field of biotechnology, a field of science that intertwines biology and cellular processes and technology, as the name suggests. PCR is used in forensics and medical science to create large quantities of small fragments of DNA in short amount of time. In forensics, it can be used to match DNA from crime scenes with those of DNA from suspects. In medical biology, it can be used to clone DNA into plasmids and check for genetic markers. For PCR to take place, you need polymerase enzymes, nucleotides, your sample strand of DNA to use, and primers. And this all takes place in a test tube, making this an in vitro reaction. Now the reaction itself takes place from heat. There are three steps to polymerase chain reactions. The first step is denaturation. The second step is primer annealing, and the third step is primer extension. The first step, denaturation, takes place at 96 degrees Celsius. This separates the double-stranded helix of DNA into two single strands, so primers can attach to it in the next step. This next step, primer annealing, takes place at 55 degrees Celsius, so the test tube has to be cooled. At 55 degrees Celsius, primers in the test tube can attach to the specific regions of DNA that the person would want copies of. Primers are specific to DNA locations, so they must be ordered ahead of time to the specific place that you want a copy of. The third step, primer extension, takes place at 72 degrees Celsius, so you must raise the temperature of the test tube for this. This step adds the polymerase enzymes to the mixture by connecting them to the DNA strands so they can bind nucleotides to finish the copy, because in the second step you only had the start and stop of your fragment. This fills in the middle part. These enzymes come from Thermus aquaticus bacterium because they have heat-resistant polymerase, as the human body is at a much lower temperature than the reaction taking place in the test tube. Polymerase is the enzyme that is involved in building nucleotide sequences such as mRNA in the body. Now, after the third step, you have one more DNA strand of a certain region, and I know what you might be thinking, this is only one copy, but I need a lot of copies. Well, this is one cycle. Because it takes place in a test tube and there's no exact ratios, you can keep repeating this cycle for dozens of times. The typical number is 35, and every time a cycle completes, you have the double amount of DNA copies that you had beforehand. Meaning that after 35 times, you would have 2 to the 35th amount of copies of DNA, which is in the billions. Now, of course, it's not 100% efficient, but you can see how this is a rapidly occurring process. This whole process can take 2 to 3 hours maximum. DNA polymerase chain reactions are rapidly aiding in medical advancements and forensics evidence in our world today, and I hope that's helpful.